Small intestine. Generally, intestine is quite long in mammals. It is about 22 feet long in the case of man. So it increases the area of absorption of digested food. So the small intestine is divisible into three parts: a proximal duodenum and a middle jejunum and distal ileum. Small intestine begins at pyloric valve of the stomach. Let us see the first part, duodenum. Duodenum is the smallest and first part following the pyloric sphincter and of the stomach. It is a U-shaped loop containing pancreas. The wall of the duodenum contains Brunner's glands, and it is a characteristic feature of duodenum. And these Brunner's glands, they produce alkaline mucus. Brunner's glands are present in submucosa. Their secretion mainly contain mucus and with little enzyme. So let us look at this in the diagram. Actually, this is the stomach part. The pyloric part of the stomach is in touch with the duodenum, and in the loop of the duodenum there is a pancreas, and along with the liver, a bile duct and a pancreatic duct opens into the duodenum. So duodenum is a continuation of the pyloric part of the stomach. Behind the duodenum, it is the jejunum. It is the middle part of the small intestine. Let us see this in this diagram. A jejunum is long coiled part of the small intestine. Generally, after the death, jejunum is empty in a dead body. This part of the small intestine is without food after the death. That is why it is called jejunum. Ileum. It is the distal part of the small intestine. It is highly coiled. The lining of the small intestine bears a series of transverse folds. They are called plicae circularis. They are also called walls of Kerckering. Then ileum is connected to the large intestine and it is guarded by ileocecal valve. It is also called ileocecal sphincter. Let us see the histology of small intestine. The wall of this small intestine possesses four basic layers, namely serosa, muscularis layer, submucosa and mucosa. Let us see the histology of small intestine. Serosa. It is the outermost layer and it is made up of thin mesothelial cells. Next layer inner to this is muscularis layer. Muscularis layer or muscularis externa is formed by smooth muscles. They are arranged in two layers, an outer longitudinal muscles and inner circular muscles. Submucosa. Submucosa is formed by loose connective tissue. This one is submucosa and it contains nerves, blood vessels and lymph vessels. You can see here arteries, veins and also lymphatic vessels of submucosa. Generally, submucosa in small intestine, especially duodenum, it contains Brunner's glands. As we have already seen, it produces mucus and little amount of enzymes. Mucosa, innermost layer lining the lumen of the alimentary canal. So the mucosa of small intestine, it forms small finger-like folds called villi. Columnar epithelial cells that lines the villi produce small microscopic projections called microvilli. So here you can see the small mucosal epithelium is folded to form finger-like projections called villi, and the base of the villi there is a folding. This is called crypts of villi. So let us see the histology of villi. As we have already studied that villi are foldings of columnar epithelium. So villi are small folds of mucosa of epithelium, and between the two villi there are small pits. These are called crypts. Generally, crypts contain endocrine cells and other secretory cells. Microvilli. So columnar epithelial cells that lines the villi produce numerous microscopic projections. These are called microvilli. They increase the area of absorption. It gives a brush border appearance. Crypts of Lieberkahn. In between the villi, The mucous membrane of the small intestine is folded, forming intestinal glands. These are called crypts of Leberkan. So crypts of Leberkan, they secrete intestinal juice that is called Saccus entericus. It is otherwise called intestinal juice. Generally, villi, they are more numerous and large in number in the posterior part of the small intestine than in the anterior part, because there is a more digested food in the posterior part rather than anterior part. Let us see the cells of intestinal glands. The epithelium that lines the villi and also crypt shows four types of cells, that is absorptive cells. They contain brush border, that is why they are also called brush border cells. Because of microvilli, they increase the area of absorption. The next one is goblet cells. Goblet cells produce mucus that protect the inner lining of the small intestine from the action of digestive juices. Next one is enteroendocrine cells. They are present at the base of the crypt. The projected part is villus. The invaginated part is crypt. And these enteroendocrine cells, they produce the intestinal juice. And next variety of cells is Penneth cells. Generally, Penneth cells, they are more in duodenum. Let us see the intestinal glands and their secretions. The epithelium of the small intestine is folded to form villi. Between the two villi, the depression is called crypts. Crypts are also lined by epithelial cells, and gland cells are scattered among them. And the gland cells of crypts are called crypts of Leiberkampf. Mainly, these secrete intestinal juice, otherwise called Saccus entericus. Whereas Penneth cells, they protect the stem cells and also secrete lysozymes. Lysozymes are antibacterial in action, and they break the bacterial cell wall that have entered into the food. The enteroendocrine cells in the epithelium, they secrete cholecystokinin, secretin, and gastric inhibitory peptide. Gastric inhibitory peptide is also called enterogastron. Whereas the microvilli, which are lined by the epithelium, contain absorptive cells. They contain small foldings called microvilli, main site for nutrient absorption. The digested food material from the intestinal lumen, they pass through the epithelium of microvilli, then absorbed into the cell. Whereas goblet cells, they produce the mucus which lines the epithelium towards the lumen, and it protects the epithelial cells of the villi from the action of enzymes. Small intestine is divided into three regions: duodenum, jejunum, and ileum. Duodenum is the shortest region, and it is a retroperitoneal part of the intestine. It starts at pyloric sphincter and extend up to 25 centimeters, that is 12 inches, and merge with the jejunum. Jejunum is about 1 meter long and it extends up to the ileum. Jejunum means empty. It is empty in dead person. The longest part of the small intestine is called ileum, and it is about 2 meters long, that is 6 feet, and it joined and merged with the large intestine, and it is guarded by a ileocecal sphincter. An ileocecal sphincter prevents the backward flow of fecal matter into the small intestine. So tears of small intestine shows all basic four layers. They are serosa, muscularis layer, submucosa, and mucosa. Mucosa, the glandular epithelium of mucosa, is folded to form crypts of Leiberkampf. This epithelium shows several intestinal gland cells. This includes absorptive cells. They contain microvilli. They increase the area of absorption. They absorb the digested food from the lumen of the small intestine into the absorptive cells and finally into the blood or lacteals. Goblet cells, they secrete mucus. This will protect the wall of the epithelium from the digestive action of the enzymes. Enteroendocrine cells are also called S cells. They produce hormones like secretin and cholecystokinin, and they influence pancreas and the functioning of the liver. Penneth cells, enteroendocrine cells, are divided into three types. One is S cells, which secrete secretin. K cells produce cholecystokinin, and other type of cells, they produce glucose-dependent insulinotropic peptide, otherwise called gastric inhibitory peptide. Pennant cells. They produce lysozymes which regulate the microbial population in the small intestine. Recently, they have discovered that they also control the functioning of stem cells of small intestine. Pennant cells produce lysozymes which kill the bacteria in the small intestine. Generally, it kills those bacteria that has escaped the gastric juice action. And Pennant cells are believed to play an important role in protecting the stem cells. Stem cells are present at the base of crypts of Labarcon, at the base of villi, and they are protected by Pennant cells.